Good evening, everyone. You're very welcome to Waterford Institute of Technology. The South East Region of Engineers Ireland, along with the Biomedical Division, are delighted to host Dr. James Kennedy this evening. My name is Mary Doyle-Kent. I'm the Secretary of the South East Region. We also have Susan Gallagher, Vice Chair, and other members of the committee, Dr. Ken Thomas, Michael McCarthy, and Owen Gill here with us this evening. From the Biomedical Division of Engineers Ireland, we're delighted to have Mark Kelleher. So I'd like to bring to your attention the fire exits. First of all, we have a fire exit here at the back, your left-hand side, and we have one here at the front, your left-hand side also. I'd just like also to bring to your attention that this talk is being live streamed on the Engineers Ireland website. So is there anybody here that doesn't want to be in the recording? Anybody? If you don't want to be in the recording, I'd like you to maybe sit at the back and don't ask any questions, because the questions are being recorded also. Okay. In terms of social media, anybody that knows me knows that I love my Facebook and Twitter. So I'd be delighted if you'd tweet us. So hashtag Biomedical Engineering, and also at Waterford IT, at EngTechWit, and of course at Engineers Ireland. That'd be super. Thanks very much. So we'll kick off then with Dr. Austin Coffey. He's one of our own here at WIT, leading the Materials and Biomedical Research Centre through the Convergent Technologies Research Group in the School of Engineering. He's also an adjunct professor of engineering in the Thammasat University in Thailand. Added to this, he is currently the president of the European Medical Polymers Association, former president of divisions of International Society of Plastics Engineer, and is currently a board member of the US Medical Devices Division of the Society of Plastics Engineering. Dr. Coffey is widely published and is very active in consultancy in the biomedical community from Boston to Bangkok. So welcome, Austin. Thank you very much.
Thank you very much, Mary, for that kind introduction. And really the function I have here is perhaps just to set the scene for Dr. James Kennedy, who has kindly come down to Waterford Institute of Technology to give his insights into the biomedical sector and also some aspects of tax credits and commercialisation opportunities. First off, I'd like to thank Mary Doyle-Kent from WIT, in with Mark Kelleher for organising this event, and also from an official capacity from Engineers Ireland to recognise the presence of Susan Gallagher, who is the Vice Chair of the South East Region. Worldwide, the medical devices sector is seeing a growth of 10% sales year on year, with SMEs, small to medium enterprises, reporting increases of sale of 15% year on year. In Ireland, in Europe, and in the US, we actually are seeing an increase much further than was originally anticipated due to, compared to emerging economies, due to political uncertainties that are currently at present, and also in terms of regulatory concerns and regulatory issues. However, technology convergence continues to be the biggest driver for innovation in the biomedical and medical devices sector. This is the bridging of all branches of engineering and science: electronics, materials, mechanical engineering, communications technologies, along with your toxicological and microbial surfaces and all this kind of stuff. This is what is giving the new opportunities, new leading edge opportunities for us. Material engineering itself, of course, towards smart and responsive systems remains the key research focus, encompassing new kind of bright new technologies, 3D printing, smart tissue engineering applications. In Europe, the industry is worth over €100 billion. 95% of companies in Europe are small to medium enterprises in the medical devices sector. 15 in Ireland, we have 15 of the top 20 world medical device companies located here in Ireland, with more than 50% of those having dedicated R&D facilities. In terms of overall revenue to the Irish economy, we are talking about €12.6 billion, representing more than 10% of Ireland's exports. There are 29,000 people directly employed in this sector in Ireland. So it is incumbent upon us to keep the research, to keep the education, and to keep the community together in events such as this in order to enhance the networking potential so that we can actually keep driving our industry. With those surprisingly fantastic stats that I just gave out, it is also quite important to mention that 50% of all Irish companies in the medical devices sector are indigenously grown. So there are huge opportunities. It's my pleasure to introduce Dr. James Kennedy. James is a Fellow of Engineers Ireland and, of course, a Chartered Engineer. Former positions included Director of the Centre of Industrial Services and Design at Athlone Institute of Technology, where he was also a Senior Research Fellow and supervised many PhD students to completion. James was Treasurer of the European Medical Polymers Division of the Society of Plastics Engineers. Prior to arriving to his current position, he was R&D Tax Manager at Mazars Ireland, a global audit, accounting and consulting group. James was headhunted to lead a new startup company, which is now a pioneer of new microbial coatings technologies, where the coating has a proven record of 99.99% kill rate of superbugs. The company, Kastus, has won the Irish Times Innovation Award 2017 in the healthcare sector, and also overall award over all business sectors in Ireland. It's my pleasure again to invite Dr. James Kennedy to the podium and to give his insights into the biomedical sector and tax credits in Ireland. As a final point, there will be a brief Q&A after James's talk. Prior to the Q&A, Mr. Mark Kelleher from the Biomedical Division of Engineers Ireland will also make a couple of words to you as well. Thank you very much.
Hello, thanks very much for having me here today. So I'd like to thank Austin and Mary. My apologies, last week I should have been here, but I was travelling. So for that, I'm sorry. I know there was a bit of organisation that had to be done around that. So what I'll do is I'll try and give you an overview of what has happened, I suppose, in terms of some of my technologies. And there's about 3 kind of presentations here, and I've kind of mixed them together. And I'm, as I said, from an engineering background, so I prefer to have some sort of questions, good questions on that at the end, or fairly open. If you want to ask a question throughout it, by all means, put your hand up and we can get through it. So as I said, I'll just give you a brief introduction to my new company that I've started to work in called Kastus. Then I'm going to give kind of a case study into a medical device that I designed when I was an academic and the process that I brought through from that. And then I'll finish up by talking about R&D tax credits, but I can probably see a lot of you here have really no interest in that. You're probably from student backgrounds, but I think it's important for you to get an understanding on the financial side of things as well. And I know for the people that's online, maybe that might be a bit more of interest as well in terms of my experience from that angle. So I'm just going to begin to give you a little background into antimicrobial resistance. You've all heard about superbugs and the way they change and adapt. Now, I'm not from a biological background myself, but what we can see, there's certain stats that there's 25,000 people in Europe dies a year, and similar figures in the US. And they estimate it by about 2050, they're looking at about 10 million lives a year are probably lost through these superbugs and the resistance that takes place. So where we come into place in terms of a company is to look at developing in terms of the surfaces and how you can kill off the bacteria from touch. And so from tiles, glass, anything at all, metals is where we position ourselves in terms of the formulation. So I'll just give a little background. The company was founded in 2015 by our CEO, John Brown. And it's a smart, multifunctional photocatalytic coating. I'll talk a little bit about that to give you an idea if you've not done chemistry before. So this has taken 10 years in development. So it was developed by a number of different scientists in DIT. And John came on board a couple of years ago and was introduced to this technology and spent years getting it up to the level where patents, a lot of money. And so I joined the company in 2017 and I was introduced to a beaker solution. And from that we've taken it then to, we're pretty much at 18 countries at the moment, global trials all around the place. So there's a lot of traveling. Just the kind of the countries in the yellow there is where we're currently at over the last couple of months. So we've been very, very busy. There's a huge demand for the product. There's a huge demand for the awareness of antimicrobial resistance as well and how prevention. I'll show you some applications of that later on. So Austin has gone through that. So to give you a bit of principles of the technology, so fundamentally it's based on a number of different chemistries. And one of the core components is the titanium dioxide. And in particular, there's 3 different states of a crystalline structure. You've probably done some crystalline kinetics or understand crystallinity from your courses, would you? In terms of, yeah, I've gone into it. So the interesting thing, these crystalline structures all kind of occur at different temperatures. But the key one that we're interested in is the anatase form of the titanium dioxide that forms within this particular product. And to give you an idea what the product really is, it's a solution. So we spray the solution onto glass or ceramics before they're fired. So as it's been heated up, the chemistries start to work and you get this crystalline kinetics taking place and this formulation of these structures. And I'll just give you a little idea how this works. So the physics works on the basis that there's a valency band and a conductive band. And you get electrons moving as a result of light coming in. So kind of from this side here, if you've got UV or natural light or visible light, wavelengths are coming in, they hit the surface, and it activates the surface to give OH and O- radicals. These radicals then can attach themselves either to different forms of bacteria, fundamentally kind of starves them, or there's other added benefits that you get a more hydrophilic based surface because of the kind of the case of the trapment of water and clean air technology, so it takes the toxins out of the air. So fundamentally that's kind of just a couple of little diagrams there to show the kind of the radicals and the process of the technology itself. Now we've bio people on our team, microbiologists and so on. When they're talking about all the different bacteria and that, this is what they're on about. I, from an engineering background, this is all I see. So when you're hearing about little bugs and running in through things, these little fellas are running around and we're trying to stop them. But it does get complex, and this is from, it's a bit disgusting, this is a mobile phone glass. And this was taken from a Forbes report that was carried out, I think, 2015. So they reckon that your mobile phone, some studies shows 5 or 30 times more bacteria than a toilet seat. So you can see the different bacteria is grown on your mobile phone screen. So the principle of our technology is, in one case, you coat the mobile phone screen, and with light, or we've other kind of components built into it, it kills off the bacteria. So you can see by the principles over on this side, it's hard to see, but you can get cell lysis taking place. Now this is just for graphical purposes, but in principle, this is one of the markets that we're looking at in terms of the mobile phone touchscreen applications. So that'll give you an idea. The other thing is, as I said, it makes it super hydrophilic, so we can reduce the contact angles and that's self-cleaning. So the properties of titanium dioxide with light, self-cleaning, air cleaning, anti-odor, and the antibacterial. So we have a solution that fits an awful amount of problems, and we're tailoring it to see what way we can, use in different industries like your ceramics, sanitary ware, and glass. So that was kind of a nutshell of the company I'm currently in. I'm happy to ask or answer any questions at the end. So what I'm going to do now, this is work. I've come from, my main background was in plastic engineering, then polymer engineering, and earlier on in my career I got involved in biomedical engineering. So my first diploma project was in tissue engineering, looking at phase inversion of polymers, and then my degree projects was looking at coatings for catheter systems, and that led me on to looking at, for the PhD, drug delivery solutions for stents. So I have a natural affiliation, I suppose, back in for medical device and the pharma industry, where I've kind of previously worked. So one of the areas that I did, as Austin, I'd say I've used an awful amount of projects down through the past, but this was one of my pet projects where I damaged my own knee playing sports. And it got me thinking about what can we do to replace, or what can we do to look at this particular area. So one of the areas I was looking at, torn cartilage. Have you heard of torn cartilage? And what happens? So basically it's down to the meniscus in your knee. And the meniscus acts as a, if you would think of it as engineers, it's a load-bearing material. It has to be able to take the resistance, the impact. You've got joint stability because your knee can turn different ways, so we've got torsional properties. It has to have a certain amount of lubrication so it doesn't dry up. You're not getting that grind and stiffness. Shock absorption. And all of these properties are built in. So it's a fantastic engineering property. It's a fantastic engineering device that evolution has created within our own knees. But from sports or wherever else, you can get that torn cartilage that takes place. So there's a couple of gruesome pictures here, not these ones, it gets worse. But what we have, I don't know if you can see here, you get these little tears, so there's different tears. And so here's the meniscus down here, it's two parts. So traditional ways of probably solving some of these if it gets really bad is to replace the knee with an artificial kind of knee joint. So this is probably an older picture of what one would look like. But as you can see, it's very heavy metal. And the next picture is disgusting. If you're a bit wary, turn away now. This is the inside of a knee operation. So you can see the amount of work that needs to take place to replace the actual knee itself. So it's quite a long process, quite a challenging process in terms for the surgeons. So these are more gruesome pictures. So the challenges on this particular project was that there was 1.4 meniscal repair surgeries in Europe and the US each year. So what the surgeon tends to do, and I remember calling into the Galway clinic and we spent a whole day looking at surgeries taking place, where they go in with keyhole surgery. So this is a good meniscus. This is the keyhole surgery here. This is a good meniscus, and this is a torn one. And what tends to happen, they go in with the kind of, assumes almost like a drill, and they cut out the crack, cut out the tear. And over time, you still might get arthritis and that taking place within the knee. It's a quick fix in a lot of ways. It's not going to repair itself. Now, in terms of, this is kind of one that's been cut open, and here's other repairs. So this one here is more of a stitch, and then there's other ones where they replace probably allograft or kind of donor meniscus into the body itself. So the breakdown of the markets was about 1.1 million for the top one, the kind of keyhole surgery, and 280k and less than 5,000 for the other ones. So the challenge for me was to look at what sort of materials can we build in to create these meniscal devices. And I had a strong background in hydrogels and looking at the effects of hydrogels from previous work we looked at. So the hydrogels are fantastic materials. You can adapt them into so many different forms, and you can make them very resistant. You can create them into drug delivery devices and so on. So I looked at particular different hydrogel technologies based on basically trial and error and a bit of knowledge from other systems to see what we could use to build an artificial meniscus. So putting this all together, this is a very busy diagram, but it's something you need to be aware of if you are designing, and probably the people in the industry are well aware of this, but it is a bit kind of clouded, but the technology readiness levels 1 to 4. So trying to build up then from the initial formulation characterization working through the samples, to verification and design, pre-production and pre-diligence. While working on a platform of biocompatibility, toxicity, clinicians, animal trials, sterilization, all the materials properties, all the regulatory, all the design, and feeding all of that kind of back into kind of, you know, marketing needs, reg approval, competitions, patents, all of this stuff, all the stuff that's carried out in the industry by a number of different people, a number of different even companies, that this is the type of stuff you need to be kind of thinking about. You don't tend to, when you're in an academic world as such, unless you're moving towards commercialization and development of the product systems themselves. So probably from a lot of your own projects, it really is lab-based and building and testing, and that's the building blocks of where we kind of go with. So in this particular project, pretty much it was 4 or 5 years, and there was about 140,000 in funding from SFI and EI, and if we were in industry, it'd probably be a lot quicker, but we're running from an academic position, so there was just time and then just getting students and getting grants to build up. So in the meantime, we were protecting the technology and working on building it as we were going. So realistically, we're looking at about 140,000 in. As I said, in industry, that would be much more multiples, but the time would be a lot reduced down. But this just gives you an idea of where we're standing on this project. So to give you an idea, and I'll kind of go through this fairly quick, some of the science that took place looking to do with the clinical formulations, what type of hydrogel systems we're using, different variations, swelling studies, thermal analysis. And we ran through a number of different iterations, and we found pretty much this version was a kind of a worked. And it gave the initial, not 100%, but we knew we could work within that range. So taking that sample, we started looking at the ability to swell, because the worst thing you could have is a hydrogel and it's known to swell. But if you put something in your knee and with all the liquids or solution that's there and it starts to swell, you're going to get problems with your joint. You need something that doesn't swell but can hold a certain amount of moisture, that it's heavily kind of cross-linked. So we start looking at different percentage of swellings and where we could work within the material itself. So then done some standard compression studies and cyclic studies to see what max loads. Then going through the literature to find out in real life what was the applications. So used some 3D printing technologies, and we built different molds to look at developing the polyphenol alcohol-based salt systems for the hydrogels. So built 3D dimensional to represent the knee. So this was basically put into a tensile tester, but we replicated the knee. So when we compressed down both of these here, we effectively had a knee, and we built in a reservoir system here of fluid and started to do compression. So constant compression studies that we were getting cyclic approach, so all the forces was being acted on. So from that, we ran probably about 10,000 cycles minimum to see were we getting any changes, and we weren't getting any hysteresis effects, so we knew we were onto something in that sense. Did all the biological analysis. Everything was coming fine with biocompatibility. It was done to 10993, so ticking an awful amount of boxes. The next stage was looking at verification, so we got cadaver samples. This was supplied by the School of Medicine and TCD, built it into the same models to see if we could get replication from cadaver versus the technology, and perfect. Everything was matching up. Really had a good product. Kept it a secret. Everything was going fine. So that was as I said, the makeup of probably about 3.5 years work to get to that level there in terms of the device itself. So the question being an academic is always publish or patent, and you get different pressures from different parts within the academic and the college kind of structures. But there's kind of merits in both, and depending on what your viewpoint is. In this particular case, we were looking at developing it for a patent. We knew we had something quite good. Done the commercialization, feasibility grants, figured out how much money we'd need, the marketplace, all of that type of stuff. And went for the program 214, and then bang. Out of nowhere, our patent attorneys found an abstract that was published, or that was sent out probably 2 years prior to this project as it was going on. And it ended up in a Malaysian university on their website, and it was enough to kill the project. Because because we thought we had a patent, we thought I had it protected, but this was just one of those things. And if you take any lesson from this, and believe me, I learned this the hard way. I only found out this after it happened, but if you have something, protection is key. You need to keep an eye on it and be wary of where it goes. So if you're posters, abstracts, keep an eye on what actually is there, because it could come back. So that actually killed all of that work. So obviously felt disappointed, deflated, all that, but I suppose at the time, you know, we got funding, graduated a master's student, had a PhD student working on our postdoc, 3 publications, gained new knowledge, all of that type of stuff. That was, I suppose, great in my old academic life. You could get away with that, but reality, if you were in industry, somebody would be out the door. You couldn't put up with that amount of understanding of work. But that's what I'm saying. It's very important to just keep an eye on your protection and where to go with it. So what I've learned in my entire career with all the projects we've completed in the centre is you can have all the stages possible, and if you treat it like a funnel, there'll inevitably be something that creeps through. And you may not see this. You can have all the quality systems in place, you can have all the checks, but sometimes due to external environmental issues, things might creep through. So you'll always have to be more vigilant and seeing, especially medtech and bio, that you can't allow this to happen. It needs to be captured as best you possibly can and try and prevent it. So it's good to understand the environments that the materials are going to. Sterilization is a big one. People do not factor in sterilization, or they tended not to, as part of the initial materials design. So sterilization as gamma, E-beam or ETO will have an effect on the properties of the materials as well. Could be an advantage or a disadvantage, so it's worth keeping an eye on those side of things. So that's kind of from the tech side as such. So what I'm going to do is, I'm going to bore the hell ahead of you a lot here on the tax credit, but I think it's probably an important side of it as well, that a lot of this stuff costs money. R&D is incredibly expensive, and for startup companies, for large companies, multinationals, SEMs, a good way of promoting R&D and keeping R&D up, there's different research going on in terms of the benefit to the economy and all of this, and for this knowledge economy and to grow jobs and to be kind of leaders. One way of doing this is probably through an R&D tax credit. And really what a tax credit is, if you've got this thing called qualifying expenditure. So if you're spending, we'll just take a figure, 100,000 euros in R&D, and it's direct R&D, you can get about 25,000 back. Now you can get that back to write off against your corporation tax, or you can get cash back over a 3-year period. That mightn't seem a lot, but for a lot of startup companies, or even some of the larger ones, there's benefits to the tax credit in the sense that that's money that comes back as cash that could maybe keep people's salaries going or invest in maybe new R&D directions or so on. But it's like everything in the tax system, there are certain rules and guidelines, and it is very onerous, and you do need a lot of documentation, and I'll just kind of go through some of it. So this is kind of a boring slide, but it comes from the revenue guidelines, and anyone that's involved in R&D tax credits, they only know too well the pain or the difficulty involved in it. So the key requirements, I suppose this is mainly for the people online if you're looking into it, is that the applicants must be a company, must be obviously within Irish tax law, but the key things must be systematic, investigative or experimental activities, be in a field of science or technology, must fall under kind of the remit of basic research, applied research or experimental development. The key things are though, it needs to seek a scientific or technological advancement and be involved in the resolution or scientific or technology uncertainty. And I'll give you ideas. The credits there, it's very onerous, there's a lot of text in it. So I spent 13 months in a job where we were assessing companies for R&D tax credits. So I tried to build up models. But as I kind of touched a little bit about this earlier, like I won't get into too much, but here are some of the kind of benefits for the R&D tax credit. Payroll costs, budgets, more people R&D. There's other bits and pieces. To give you an idea of the R&D environment within Ireland, and I think this is an important slide. It's very busy, so I won't go through it on. You can read the bits yourself out of it. But this is from the CSO 2017 analysis looking at R&D active companies in Ireland. This is available on the CEO website. But as you can see, like pulling certain things there, there's like 1.3 billion was spent on labour costs and R&D. There was 1,900 enterprises engaged in R&D activities in Ireland in 2015, where 73% spent less than 500,000, 1/6 spent between 500 and 2 million, and 10% spent over 2 million. So if you start thinking about it, like the amount of money that's been spent on R&D and the amount of companies, the ability of a tax credit to claim back some of that, you can feed back into the structures within the organisation. So as I said, that's quite a busy slide, but you can get it on the CSO, and it has got all the previous years as well built into it. So bringing it back to kind of an example, if we look at this product development process, and this is very generic really, to be honest, and if I take the idea that I showed you with the medical device, so if we take that it was a company and we're going ahead for the purpose of this talk this evening. So the initial stages, we've got pre-concept, evaluation, feasibility, design. Within all of those, evaluation, feasibility. So we've got all those generic building blocks, and where does the kind of terms fit in that I mentioned earlier? The R&D terms, which are pretty much common around, I suppose, all research, but revenue takes a lot from the Verscati manual as well. So there's the basic, applied, and experimental development, and then there's no claim. Realistically, if we have a graph like this, and we have initial knowledge and concept, so your initial R&D ideas, your initial what are we going to do, building up, basic research, applied research, development, and technical services, and then the final product. To be honest, in Ireland, in most cases, it's probably really a lot of small R and a big D, a lot of development. You get elements of R, I'm not discrediting the R is incredibly important, but especially a lot kind of, I suppose, multinationals, depending on where you are, but that tends to be the kind of mix in some cases. For tax purposes, you're really within that window of what is R and what is D and what you can claim for in terms of the credit. And if you're involved in it, it's important to talk to a professional service firm to give you an idea, because sometimes they keep changing the regulations or what you can or can't claim for in terms of qualifying activities. But certain areas, what as a researcher or even academic with classes R&D is not classified or allowed under revenues terms. So like fundamentals like travels or just some sort of pre-concept designs or all of those sort of things. So it's important to have a good understanding. And I'll try and give you, to the best of my knowledge, a guide, and you can see for yourselves if you're going to use it in your own industry. So there's two tests you need for R&D tax credits. One is called the science test, and the other one is called the accounting test. So that original picture I showed you earlier on of revenue guidelines, the engineer tried to put it into a visual, into a model to make it easier. And this is a complement of when I worked in Mazars. So the key thing is to build an advance in the overall knowledge or capability in the field of science or technology. You can only claim for the 12 months. So if your project's 3 years, you can claim for that 12 months that you worked within that year, the academic year, or financial year. Work falls under basic research, applied research, or experimental development. So in this case, I just picked applied research. You have to be able to show that there's a scientific or technological advancement. What you're trying to do has got something that's not been done really before, and you're trying to advance science or technology. And if it's not been done before, the other thing that they say, actually, I'll bring this up, just say we're trying to do something in this company, and you've got a team of engineers trying to develop it. The question is, if I have the same company over here or a different type of company, say a medical device, and they have a team of engineers, would they be able to replicate it? And this is a hard thing to kind of prove, because there's always different angles or different areas, but there's this concept called a competent professional working in the field. And so it kind of rules out that you do need your people who are working on it to have maybe the qualifications or the experience to actually do this type of stuff. And with everything that's you're trying to advance, there's always going to be uncertainties. If you knew how to do it at the start, you wouldn't go about it wouldn't be kind of the research. So you need to have these uncertainties that you're trying to solve. And that kind of fundamentally brings it back. So you can claim for as long as the uncertainties have been reached. So as soon as you reach that uncertainty, that's it, you can't run into it. So looking at the meniscus one, kind of some of the questions, has it been done before? No. Published? No. Public domain? No. So if you can do all of these kind of checks to make sure that it is new or novel or works. Now, as I said, it's quite difficult. It's much easier for the real R, but when you're over on the D side and the experimental development and you're in maybe scale-up or validation trials, it's hard to find sometimes the novelty, even though you don't know how to maybe optimize things, or there is things built in for process, but as I said, it's worth checking the guidelines just to confirm that. So kind of filling up that model, would say what was the scientific uncertainty? We'll say the elastic response from the meniscus. So we wanted to see that we were getting not too much of elasticity, that we were getting a slight elastic response. What was the uncertainties that could result from that? Leachings of the salts within the system, crystalline structure, or the elastic recovery. So you build up the documentations and the science around each of those, and then you have them resolved. Equally, there's the accountancy test. So this is really for the accountants and financial side of it. They need to be able to look at what's spent, what's allocated to the expenditure, to the R&D, and the percentages allocated to those side of things. So that's kind of built into the accountancy angle on it. Now, this, just to put it into perspective, this is some research I've done myself, looking at the effects of R&D tax credits. And what I found, and this was a combination of a number of different resources, the credits came about in 2013 or 2003. So in 2004, there was 75 companies claimed 70 million, and this is from figures in the credit. If we jump to the latest figures I could get my hand on was about 2014. What we have is from the 75 companies, it's jumped up to just under 1,600 companies claiming the credit, and about $500 million in kind of, I suppose, credit been kind of given back in its different forms. The key thing to look at is the business expenditure in R&D. So it was about 1.2 billion in 2004, and this is probably a good indication as well in terms of the economy from different credits, the innovation, and all the infrastructure that's been put in place in terms of education development, that we see that there's an increase up to about 2.1 billion. It's probably higher on the business expenditure and R&D, word value. But the key is, with all of that increase, obviously the revenue we're getting handing out about this money. So if you are doing it, you need to be aware that you have all your documentation in place, because they do hire probably people from, they used to anyway, was people from academia, and they potentially, this is published in the Irish Times pretty much 2 years ago from now, up to €1,000 a day to assess that what you're doing is actually R&D. So there is checks there now, and there is financial checks and so on. And you do a number of outcomes. Revenue approves the claim, you get installments, they may query it, or they may send you 23 questions to answer certain things based on your science and your run into it, or you might get an audit. But the key thing is, I won't go too much into this, you need documentation to back up every single thing you're claiming for R&D, and to show that. So it goes back to notebooks, just general timesheets, journals, every emails, anything you have that you can prove what you're doing is trying to resolve these uncertainties, and what you're trying to seek a scientific achievement, scientific advantage through these uncertainties. So you need to be able to back up all of this. So that's what I'm saying. It really is, a lot of companies don't claim the credits because there's a lot of paperwork and administration. Sometimes you might need a dedicated person for this type of stuff, but it's a potentially a good way of getting money back into the organisation. I won't dwell on this one too much because this will bore you, but this is a new incentive called the Knowledge Development Box. And the thing about the Knowledge Development Box, you heard of this thing called the Double Irish, and this kind of came in pretty much near enough after it. But a knowledge box it's a tax relief fundamentally on your products that's been patented. So with the R&D tax credit, you can claim on it if your technology has failed, but the knowledge box is that you're actually in sales, you're generating a profit. or you're selling it, should I say, and it falls under the OECD modified nexus, and that's pretty much this equation. Now, I'm not an accountant, so I'm just put it up there for to give you an idea. So there's different rules and regulations around each of those, so I wouldn't be qualified to talk about this. I think it's important you talk to your financial people if you are. And so I tried to build it up as a model myself. I like systems and I like structure. So the R&D tax credit, 25% qualifying activity, so you can run it through, you get a corporation tax liability or refund over 3 years. But if you set up your systems correct and you own your patents, realistically you can also avail off all the documentation. The credits feeds into the knowledge development box as such, that you get the 25% back on the expenditure, and then on your patents on qualifying activities, instead of paying 12.5% corporation tax, you can pay 6.25%. So it allows roughly about around 50%. Now, as I said, I'm not an expert on this, so you really need to, if you're interested, talk to financial personnel on all of that. So I think I'm on time, am I? Let's go up into it. And so I'd just like to thank you all. That was a quick tour, but I'm happy. I prefer to answer questions anyways, if I can at all. And so hopefully that was of some benefit. It'll give you some idea. So thanks very much.
State your name and affiliation, please.
Okay, my name is Arun Mathur. I'm from Bosch & Lomb. Okay, in the beginning you said something about the titanium oxide coating. What is the thickness of the coating?
Depends on the application, but we're looking at maybe 50 nanometers up to about 80 nanometers would be the range that we're looking at on our products.
And how is the coating applied?
Spray.
Okay. And can it be applied to hydrophilic as well as hydrophobic surfaces?
Yeah, what we found is the hydrophobic surfaces that we're spraying on, it reduces down into hydrophilic. So if you've got a high contact angle that's nearly 70, we were finding it was getting down to about 35.
Yeah.
But if you were getting 35, 40, we could get it down under the 20s and so on. So it does have an effect. The surface energies that are taking place would have an effect on the main substrate.
And does it work only at a particular wavelength and frequency of light, or?
We've this is, I suppose, where the patent is built in. Traditional titanium dioxide system primarily works under the UV, but we've built in an element of another component that increases the wavelength, so we can get the natural and visible light built into to activate the coating system.
And does it kill only harmful bacteria, or does it kill the beneficial bacteria also?
To be honest, we work to a standard ISO 27447 2009, and that's looking at there's 1 or 2 different bacterias. So we're really targeting like the MRSAs and the other ones, yeah.
And now titanium oxide is an abrasive material. Is there any concern of harms caused to the patients by abrasion?
No, it's a good question, but we start to see those literature reviews coming out now at the moment, because titanium dioxide is used in so many different, paints, pharmaceuticals, food. It's so broad, but it is generally regarded as safe as a material itself. We've got studies sent out to show that it's from our ink coating that's non-toxic and the key thing in a lot of this stuff is non-leaching as well, because we're an alternative to silver-based compounds, where silver is getting a lot of bad press, and rightly so. It's getting into the environment, it's causing a lot of damage. So we're an alternative to silver systems.
Okay, you also spoke about hydrogels. Are these hydrogels carbon-based or silicon-based?
They're carbon-based, yeah. Well, 15, 20 years of hydrogels. Yeah.
Okay. And have you conducted clinical trials on animals or just humans? Or
in another project, I have done clinical trials, but it wasn't on a hydrogel technology. It was looking for an animal. So we've that patented at the moment, or it's gone through the trials. My old life as an academic, yeah.
Okay, thanks very much. Thank you.
Yeah. Yes, I signed up. Look into it.
Again, if you can state your name and affiliation, please.
Hello, Ulrich Stafford, Boston Scientific. My question is about the same topic because it brings me back to long distant memories of my PhD research in a similar area. But the question I would have about these type of coatings is that the technology is has been around for a while, but there have been some issues with it. One of the ones I'm kind of wondering how you've dealt with is the scratch resistance.
We've seen, no, it's a very good question. Because of the temperature variations, we'll say in ceramics, we've seen that there's been, if we're doing an indentation test, the non-coated one under a force of, I think, was it 5 centimeters, if I can remember the test, there was 100 nanometers of a scratch. The coated sample, we were getting a scratch down to 10, so we were seeing about a 10 times improvement. Now with glass, it's probably 50% of those, because glass is a thinner coating on the sample. The key is, it's not just titanium dioxide we have in the formulation, I suppose. We've other components built in, and from our industry trials we're finding that's actually improving a lot the glass and scratch resistance, and there's a lot of mechanical properties increased as well. So it actually turns into a multifunctional approach. But you're right, traditional, I suppose, the titanium-based oxide ones, but this is a spray chemistry. There's other chemistries activating in it. Any project questions from the students? If there's any things you might be caught on if you are doing any 4th year or 5th year projects, or in terms of materials, or all shy.
Okay. Yeah, maybe if I can invite Mark Kennedy up to the podium. Thank you very much, James. Sorry, Mark.
That's okay, that's just gone, Austin.
Can you please ensure that anybody who is here has signed the attendance sheet in terms of Engineers Ireland and WIT? So whoever hasn't, just please put their hand up.
Thanks, Austin. Hi, everyone. Tonight's event is part of a series or a program of events jointly organised by the Biomedical Engineering Division of Engineers Ireland, various regions within Engineers Ireland, the Institutes of Technology and universities. So I'd just like to bring to your attention a couple of further events which are happening in the next couple of months. In the next few months, on the 2nd of February in St. Vincent's Hospital, there's a day of shared learning, which is the Irish Posture and Mobility Network. Speakers from a variety of fields will talk on a wide range of topics related to innovation, research and best practice in seating, posture and mobility. The intended audience there is our nurses, doctors, occupational therapists, clinical and rehabilitation engineers and technicians, product design and designers and product specialists. On the 27th of February in NUI Galway, it's a presentation on the needs-based medical device innovation, which is a personal account of the BioInnovate Ireland programme. And the presenter there is Dr. Andrew Cameron. On the 20th of March in IT Tralee, the presentation on next-generation medical implants, opportunities and challenges in orthobiologics. And the presenter there is Dr. John Gleeson, who's the CTO for Surgical Technologies Limited. These are confirmed events. We have a couple more events in planning as well, and hopefully get those finalised in the next couple of weeks. In February in IT Tala, we have a planned event. It's going to be in the theme of a surgical and clinical area. And then in April in Galway, we have a planned event. It's a one-day seminar. It's excellence in manufacturing medical devices. We're hoping to get industry leaders from medical device manufacturers in Ireland to speak and to develop and work through some case studies in manufacturing excellence and process excellence. Details of all of these are on the Engineers Ireland website. Go to the website, click on events, and you'll see them published there, all details there as they go through. Finally, a bit of a plug for the Biomedical Engineering Division. We have our own page on the Engineers Ireland website as well. Click on groups and then on divisions. But if you have any questions on who we are and what we do, or if you have any ideas for events we could run, if you want to get involved, I'd ask you please feel free to come up and talk to me after the event. I'll be glad to speak to you. Okay, thank you.
So just finally, Thank you all very much, both out in cyberspace, and thank you for those of you that are in WIT this evening. We're delighted to see so much interest in biomedical engineering. And Susan, the Vice Chair of the South East Region of Engineers Ireland, would like to make a small presentation to James. So, Susan, would you like to come forward? Thanks, everybody, and have a lovely evening and safe home. Bye-bye.